Okay, so we've got a fair amount to do to finish up the term. Surprise, surprise. We have a fair amount to do every day, right? So nothing new in that. Today I want to finish up the last of the material relative to the citric acid cycle. Shh. Okay? I want to finish up the last of the material relative to the citric acid cycle, and that means coverage of another cycle which is actually related to the citric acid cycle called the glyoxylate cycle. The glyoxylate cycle is an important cycle, particularly in plants and bacteria. We do not have this cycle within us, and as you will see, it overlaps with a fair amount of the citric acid cycle. The reason we can't do it is we lack a couple of the enzymes that this cycle has. So here's what it looks like. I've drawn it out schematically where we imagine we start with citrate, which is what we would get in the citric acid cycle if we put together acetyl-CoA and oxaloacetate. And that's how the glyoxylate cycle also starts. Okay? Citrate's converted to isocitrate, same enzyme as the citric acid cycle. But here's where the pathway differs. Instead of isocitrate becoming decarboxylated, isocitrate gets split into two pieces. And those two pieces include a 2-carbon molecule called glyoxylate and a 4-carbon molecule, succinate. Well, succinate, as you know, can be metabolized in the citric acid cycle, and so it is. It's occurring in the same place. So the same enzymes of the citric acid cycle are there, and the succinate turns into fumarate and turns into malate. Okay? The glyoxylate, however, provides another opportunity. So what we're doing in the glyoxylate cycle is we're bypassing the decarboxylation steps of the citric acid cycle. So you may recall there's 2 decarboxylations in the citric acid cycle, each one giving off a carbon dioxide. In the glyoxylate cycle, we bypass that, not we, but plants and bacteria bypass that, those decarboxylation steps so that they keep those carbons. Well, that becomes important because glyoxylate can be combined. Oh, by the way, the first enzyme that does that is an enzyme called isocitrate lyase. That's what it's called. That is one of the 2 enzymes that we don't have. So this is one of the reasons we can't run this cycle. But plants and bacteria have this enzyme, isocitrate lyase. The glyoxylate can be converted into malate by combining with another acetyl-CoA. And the second enzyme that's necessary for that is called malate synthase. So if you're keeping track, what we've done is we started with a 4-carbon piece, oxaloacetate. We added a 2-carbon piece in the form of acetyl-CoA. We broke off a 2-carbon piece, and then we added another acetyl-CoA. So we have input 4 carbons into this cycle, and we've lost none. Meaning that at this point, we actually have 2 molecules of malate. In the citric acid cycle, we would only have 1. Okay? Well, that's important because 2 molecules of malate get converted into 2 molecules of oxaloacetate. And if I'm in the citric acid cycle, I have to use that oxaloacetate to keep the cycle going. Because if I take away my oxaloacetate, the cycle will stop. But here I've got an extra one. Every time the cycle turns, I've got an extra oxaloacetate that's not doing anything. That extra oxaloacetate can go off to processes like gluconeogenesis. Now, that may not seem like a significant thing to you, but it turns out to be a pretty significant thing. What plants and bacteria are doing is that they're taking acetyl-CoA and they can make glucose directly from it in net amounts. If we try to do that, we stop the citric acid cycle and we don't get any more oxaloacetate. So plants and bacteria can make glucose from acetyl-CoA in net amounts, and we can't do that. If we could do that, it would be a really good way to burn fat to make sugar. But we can't do that. I think about that as I'm watching my waistline. watching it grow and grow and grow. That's what I'm, bad joke. Yes? So it's making 2 oxaloacetates. We've put 4 carbons in the form of 2 acetyl-CoAs, and we haven't lost any. So we end up with an extra oxaloacetate. Okay? Now people say, well, when does the citric acid cycle run and when does the glyoxylate cycle run? And the answer to that question is whenever it's convenient or needed for the cell. Okay? This cycle bypasses decarboxylations. And what do decarboxylations require? They require NAD. If the cell has a lot of NADH, this would be a good way to go because NADH is needed to make glucose. Okay? Make sense? Now, later when I talk about photosynthesis, I'm going to remind you about glyoxylate. And the reason I'll remind you about glyoxylate is because glyoxylate is a toxic substance. But plants have evolved a neat way of dealing with that toxic substance. And we'll talk about that later. Okay, questions about that? It's basically due to the fact that these plants and bacteria have these 2 enzymes, isocitrate lyase and malate synthase, that allows them to do what you see here. Okay. Well, that's the glyoxylate cycle, and now we get to turn our attention to electron transport and oxidative phosphorylation. The lecture today is the sort of meat of metabolism. This is where everything comes together. Okay? This is where we begin to understand phenomena such as respiratory control. Respiratory control. All right? We start to learn better how our body is responding to the environment that we put it into, whether it's exercise, starvation, lack of oxygen, etc., etc. We better understand what's happening as a result of, hopefully, the lecture today. Well, I'll remind you of this figure I showed you the other day, which is the structure of the mitochondrion, and I'll point out several of the features to you. I pointed out earlier that the most important part of it was the inner mitochondrial membrane, and that's these invaginations that you see right here. It's in the inner mitochondrial membrane where the electron transport and oxidative phosphorylation enzymes are found. Okay? That's where they're found. There's also an outer mitochondrial membrane, and it doesn't do an awful lot. Okay? The inner mitochondrial membrane is much more important because if you poke a hole in the inner mitochondrial membrane, the mitochondrion will burn itself up. Okay? So keeping that inner mitochondrial membrane intact is very important for that mitochondrion to function and ultimately for the cell to function. These infoldings of the inner membrane that you can see here that I talked about earlier, I didn't give them a name, but I'll give them a name here, and that's known as cristae, C-R-I-S-T-A-E. Cristae are just the infoldings of the inner mitochondrial membrane that's there. Okay. Well, let's talk about the electron transport system. Here's my confusing figure for the electron transport system. Before I tell you about the confusing figure, and by the way, I'm not going to tell you hardly anything about the confusing figure because I don't like it. All right? I need to tell you what's happening in electron transport. Electron transport is just that. It's a system in the inner membrane of the mitochondrion where electrons are being taken from either NADH or FADH2. And when they're taken from NADH or FADH2, that leaves behind NAD and FAD. So this is how the cell is regenerating NAD and FAD. Well, in order for the system to take electrons from those two, the system has to be running. And what does it take to run the system? Well, as we will see, oxygen. Oxygen is necessary to run the system. That's what I've been talking about before when I say things start to ferment because they run out of oxygen. When they run out of oxygen, they can't do electron transport. And when they can't do electron transport, NADH concentration goes up, FADH2 concentration goes up, and then glycolysis and things want to stop. Okay? All right. Well, this is a confusing figure. It shows the entry of electrons from NADH and FADH2, and it's got a bunch of blah, 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 and I don't like it. So we're not going to talk about it. Okay? Instead, we'll talk about it here, and this looks a little bit more confusing, but it's actually not, because I'm going to simplify it for you. What you see on the screen is a depiction of the inner mitochondrial membrane. There's the lipid bilayer of the inner mitochondrial membrane. You see in this membrane that there are several protein complexes and individual proteins that are embedded in it. Okay? These complexes look like there's all kinds of things happening in them, and there is a lot that's happening in them, but we're going to keep it very simple. Okay? The first complex that we see on the left, okay, I call complex 1. In fact, most people call it complex 1. It has a long mouthful of a name, but if you notice complex 1, that's fine. What's the function of complex 1? Complex 1 takes electrons from NADH. It passes them through a variety of proteins, but we don't need to care about the individual movement within complex 1. And as the electrons are moving through complex 1, something interesting happens. The something interesting that happens is the movement of electrons creates an energy force that kicks protons out of the matrix and into the intermembrane space. You might say, what's the intermembrane space? You haven't told us about that. That's the space between the inner membrane and the outer membrane. A little gap in there. Okay? So protons are being kicked out of the matrix and into the intermembrane space. When complex 1 is done with the electrons, it passes the electrons off to coenzyme Q. This is a little molecule that's capable of accepting electrons. By the way, when you get electrons off of NADH, they come in pairs. Okay? So 2 electrons enter here. And coenzyme Q has a very useful property. Gesundheit, yes. The very useful property is coenzyme Q can accept electrons in pairs, but it can pass them off one at a time. That turns out to be important because the rest of the system wants electrons one at a time. So the electrons have moved through complex 1. Some protons got pumped out. We've started to create a proton gradient. And we know that a gradient can be used to do things. Coenzyme Q has accepted the electrons. And now coenzyme Q travels within that lipid bilayer and goes over and deposits them into complex 3. I'll talk about complex 2 later. So from complex 1 to coenzyme Q to complex 3. Coenzyme Q does not pump any protons. No protons pumped here. It's simply a way of shuttling electrons between complex 1 and complex 3. When the electrons get to complex 3, the same thing happens. Electrons move through complex 3. As they move through complex 3 pumps protons out of the matrix and into the intermembrane space. So again, we're building up the proton gradient. The next thing that happens in the process is those electrons have to go somewhere when they pass through complex 3, so they get dumped off onto this little tiny protein called cytochrome C. Cytochrome C is a very interesting protein into itself, and I won't tell you all the details of that, but it's a protein that's found everywhere from bacteria to humans, essentially. It's a very interesting protein. And like like coenzyme Q, cytochrome C acts as a shuttle to move electrons. In this case, it's moving electrons between complex 3 and complex 4. Cytochrome C does not pump any protons. So far, the only things that have pumped protons have been complex 1 and complex 3. Don't confuse electrons and protons. Common thing for students. Electrons are moving, protons are being pumped. Okay? When cytochrome C comes over to complex 4, which is the last destination over here on the right, it dumps the electrons into complex 4. And complex 4, just like complexes 1 and 3, pumps protons out of the matrix and into the intermembrane space. So we see that the proton concentration is growing in the intermembrane space, and it's falling in the matrix, meaning that the pH will be lower in the intermembrane space than it will be in the matrix. The electrons pass through complex 4, and they arrive at their terminal electron acceptor. We've got these electrons moving. They've got to go somewhere. And so these electrons hit their terminal electron acceptor, which is oxygen. And when they get to oxygen, oxygen is converted from molecular oxygen into 2 molecules of water. It takes 4 electrons to convert 2 molecules of O2 into 2 molecules of water. So you can see why, if the cell runs out of oxygen, what's going to happen. If the cell doesn't have a terminal electron acceptor, electrons pile up inside of complex 4, which means that cytochrome C will have no place to put electrons, which means that complex 3 will have no place to dump electrons, and you can see the system backing all the way up until we get to the point where NADH has no place to dump electrons. This is why oxygen is essential for this process to occur. If the cell does not have oxygen, it cannot convert NADH back to NAD. Yes? Okay. So we have, all right, so electrons come in pairs. They get here and they start going one at a time. Ultimately, 4 electrons, meaning 2 pairs, will come through here and convert O2 into 2 H2Os. Oop. There you go. Sure. Okay, well, what about FADH2? I haven't talked about FADH2. I just talked about NADH. It turns out FADH2 does its electrons almost identically from the coenzyme Q point to the right. In other words, electrons from FADH2 do not enter at complex 1. Electrons from FADH2 enter from something called complex 2. And then complex 2 dumps electrons off to coenzyme Q, and the process you've already seen continues to occur. Okay? Now you might note that electrons coming from FADH2 are bypassing complex 1, which means that there isn't going to be as many, or there aren't going to be as many protons pumped for electrons from FADH2 as there is compared to NADH. Okay? As we will see when we get to the next phase, which is oxidative phosphorylation, there are enough protons pumped to generate approximately 3 ATPs for electrons coming from NADH and 2 ATPs for electrons coming from FADH2. People argue over exactly how many there is, and it doesn't really matter. It's approximately 3, and it's approximately 2. Okay. Now, the one thing up here I haven't mentioned is this thing here called the Q cycle. I'm not going to show you the Q cycle, but I will tell you that the Q cycle is something that's involved in moving electrons from coenzyme Q into complex 3. You'll also notice up here that there are a lot of atoms of iron that you see on the screen. Iron turns out to be a really, really important atom for transferring electrons because iron can exist as a +2 form and also in a +3 form. And what that means is that the difference is 1 electron. Moving electrons simply shuffles the oxidative state from iron from +3 to +2 to +3 to +2 to +3 to +2, back and forth. Okay? Complex 4 is a little bit different than the others in that in addition to having iron, it also has an atom of copper in it. Copper. And copper also has 2 oxidative states, copper +1 and copper +2. And yes, those are involved in the transfer of electrons. Yes. Okay. Questions about that? Okay. Let's see. You guys really want to get confused? There you go. Okay, we're not going to do that. Coenzyme Q, in contrast to cytochrome C, is a small molecule. It's a small molecule that can exist in 2 different oxidative states. And I'm not going to ask you to memorize the structure or anything like that. It's actually 3 oxidative states, I should say. All right? And that existing in 3 oxidative states allows it to pass electrons off in 2 different ways. So it can pass electrons one at a time. It accepts them in pairs and passes them off one at a time. Okay. Well, we've just done what I like to describe as charging the battery. When you charge a battery, what do? You're putting power into a little device that's going to provide that power for you when you need it. The battery in your car kicks in because it's been charged before you turn the car off, so that when you turn the car back on, the juice is there to turn the starter and get the car going. We've charged the battery here by pumping protons. Protons got pumped out. We have a gradient now. We have more protons outside the inner membrane than we do inside of the inner membrane in the matrix. The cell will use that gradient to make ATP. Okay? And here's how it does it. There's a complex that we have in our cells, and this is true for all cells, okay, that has a very fascinating collection of proteins that are referred to by a couple of names. Okay? Most commonly, it's called ATP synthase, ATP synthase. But it's also called complex 5. Complex 5 will confuse you because when you say complex 5, you will think it's linked to complex 4 or electron transport, and it's not. But this ATP synthase that I'm going to describe to you is a remarkable protein. This remarkable protein allows protons to run through it. And when protons run through it, this little guy at the end, or a thing inside this little guy at the end, spins. Just like a turbine at the bottom of a dam spins and generates electricity, there's something inside of this head of this mushroom structure that spins, and when it spins, this guy makes ATP. It's a molecular motor, the likes of which are remarkable. This is found in the membrane of our inner mitochondrial membrane. Okay? Like the electron transport proteins, this is found in the inner membrane of our mitochondrion. Okay? And there's a very similar type protein found in the chloroplast of plants. Meaning that the chloroplast of plants generates ATP in photosynthesis in a very similar fashion to the way our mitochondria generate ATP. This is a schematic look at this protein. Okay? You can see the membrane, all right? And you can see up here this mixture of subunits above. And the part that does the rotating is this little guy in the middle right here. This little guy in the middle rotates, and when he rotates, what it does is it changes very slightly the configuration of the proteins up here that are holding onto ADP and phosphate. And as a consequence, ADP and phosphate get squeezed together, and ATP gets made. All right, let's see how that actually occurs. Okay? Not what I want. All right. This is looking at that complex of proteins from the top down. So we're looking at the top of the head of that mushroom, looking down on it. And it turns out that the proteins that are in here are 3 identical proteins. But their shape changes very slightly depending upon how the thing in the middle of them, which isn't shown here, is positioned. Okay? It depends on how that's positioned. As we look at this, we see that there are 3 states. A state called L, a state called T, and a state called O. Same protein in 3 different forms, and the structure of that protein is changing. Right? So currently the top protein is in the L form. L stands for loose. Okay? In the loose state, the protein will bind ADP and phosphate. In the tight state, what the protein will do is it will squeeze together the ADP and phosphate to make ATP. And you can see that happening. Follow the L here. L is empty on the top. L has bound ADP and phosphate here. L has started to change shape, and it's squeezing together the ADP and phosphate that you can see right here. And finally, they get squeezed together, and now we're in the T form. So we've gone from the L above to the T below. As the L changed into the T over here was doing something on its own. The T had finished the last cycle with ATP, just like this guy is getting ready to do here. Except for when it finishes with the ATP, it no longer has use for it. So as the L is changing into the T is changing into, that says T, that should be O. It's changing into the O form. O means open, and open means let it go. Let it go. Okay? That should be an O at the bottom. My apologies. All right? So the ATP has been released in the open form. Well, what's happened to the open? The open started out up here, and it started changing shape so that it could become the new L. And this is where the next ADP and phosphate will come in. And on the next cycle, it just continues and continues and continues and continues. This wheel actually spins, or the thing inside of here actually spins, just like a turbine. Really remarkable. And what causes it to spin? The proton gradient, because protons are coming from outside of the inner mitochondrial membrane, that is from that intermembrane space where the concentration is high. They're leaking through this complex, causing it to spin, and they come back into the matrix. The gradient makes that happen. Okay? It's a remarkable process, an absolutely remarkable process. Okay. Now let me ask you a question. Let's imagine, if you will, that you are sitting here and somebody decides to fill the room with nitrogen. What's going to happen to your electron transport? Your electron transport is going to slow down and it's going to stop, right? When your electron transport stops, what's going to happen to the pumping of protons? It's going to stop. So the gradient isn't going to stay there forever. Eventually, the two sides are going to equalize, right? So you will stop making ATP. And when you stop making ATP, you know you're in deep doo-doo, right? You suffocate. Okay? If I stop, let's continue along about why we have problems. If I've stopped electron transport, what happens to the concentration of NADH? It goes up, right? Because no place to dump protons. As it goes up, what happens to the citric acid cycle? It stops, right? Because NAD is needed for the citric acid cycle. What happens to glycolysis? It's going to go to fermentation, right? And fermentation generates 1/19 as much energy as does when we have oxygen present. We can't last for very long without oxygen or we're hosed. Everybody with me? You could describe that to me on an exam? Okay, let's mix it up. I like to mix things up. Here's an interesting molecule. Those of you who are dietitians may have heard of it. Anybody who's ever heard of it should stay away from it. Okay? And I've actually heard of people in recent weeks who have actually been poisoned by this compound. It's called dinitrophenol, 2,4-dinitrophenol. Okay? Just call it DNP, dinitrophenol. Okay? Dinitrophenol, okay, is something we call an uncoupler. What's an uncoupler? Well, the process I've just described to you describes coupled mitochondria. Coupled meaning that electron transport creates a gradient, and the gradient drives the synthesis of ATP. The process of electron transport is coupled to oxidative phosphorylation. Understand? They're separate processes, but one feeds the other. Electron transport provides the protons necessary for oxidative phosphorylation. Oxidative phosphorylation uses the protons to create ATP. Let's talk about this guy. What does this guy do? This guy uncouples. And how does it uncouple? It uncouples in a very, very simple fashion. It pokes holes in the inner mitochondrial membrane. It pokes holes in the inner mitochondrial membrane. Well, if I poke a hole in the inner mitochondrial membrane, let's think about what would happen. NADH dumps electrons off into electron transport. Electron transport pumps protons out, but there's a hole, or a lot of holes. Normally, those protons would only come back through the ATP synthase. But now they've got an alternate way to come back in, right? What's going to happen? They're going to leak through the holes, just like if I poked a hole in the dam, it's going to go, the water's going to come out the hole in the dam, and very little of it's going to go through the turbine, right? I got a problem. What I've just lost is respiratory control. I've lost respiratory control. Okay? Let's think about what that means. Let's think I didn't poke the hole. Okay? I've got a tightly coupled mitochondria, meaning the two processes are dependent on each other. Electron transport's driving oxidative phosphorylation. If I stop electron transport, I will stop oxidative phosphorylation because the gradient runs out, right? What if I stop oxidative phosphorylation? Will I stop electron transport? What do you think? The answer is yes, when I have respiratory control. How does that happen? Well, let's imagine I'm sitting around my house. It's Memorial Day. I don't feel like mowing the yard. I don't feel like doing anything except sitting out in the backyard, drinking a beer, eating pizza, and watching the world go by. Probably the same things that you guys did, right? I'm not exercising. I'm not doing anything. I'm just sitting there, absorbing all that nature has to offer me. What's going to be my ATP levels? High or low? High, right? I'm not burning anything up. My ATP levels are high. When my ATP levels are high, what are my ADP levels like? They're low, right? What do I need for ATP synthase to work besides a proton gradient? I need ADP. Because ADP is necessary to make ATP. And if I don't have any ADP, I can't make ATP, I can't load the turbine won't turn. When I'm sitting around, let's think about this, sitting around drinking beer, eating pizza, and doing nothing but swatting flies, all right, my ADP levels are high. My proton gradient is growing, growing, growing. And eventually the proton gradient gets so high that I can't pump any more protons out. It's so high. It's like pushing water overhead, and you keep pumping water overhead, and finally the water over your head gets so immense you can't pump any more. Right? So when I'm not burning up that ATP, I will stop electron transport if I have tightly coupled mitochondria. Because the gradient becomes too large for me to overcome. All right, let's think. Let's keep thinking here then. We stop electron transport. What happens to NADH? Oh, back off here. Back off before I say that. When I stop electron transport, what happens to my need for oxygen? Right? I'm not running electron. Am I breathing heavily when I'm sitting out there in the backyard drinking beer and eating pizza? No. Right? Electron transport is stopping. When electron transport stops, what happens to the citric acid cycle? Slows down. What happens to glycolysis? Slows down. What happens to my burning of things in my body? Slows down. I'm getting fatter. Right? When I don't exercise, that's what's going to happen, guys. That's what's going to happen. However, if I get up and I decide I'm going to burn off this beer I just drank, I'm going to go for a nice run, what's going to happen? First thing that's going to happen. What's the first thing that's going to happen? ATP levels are going to happen. What's going to happen to my ATP? It's going to fall because my muscles are using ATP, right? ATP levels fall. What happens next? ATP synthase now has ADP. It starts spinning. And when it starts spinning, what happens to the proton gradient? It starts to fall because protons start coming back in. And when the proton gradient falls, what happens to electron transport? It starts. And when electron transport starts, what happens to my need for oxygen? It increases. And when my oxygen need increases, I start breathing heavily. And when electron transport is running, what happens to the citric acid cycle and glycolysis? Yeah, I'm burning things up, right? And we know from the Big Bang that there's a lot of heat generated, right? And the more I'm running that Big Bang reaction, the hotter I get. It all makes sense. It all makes sense. At least it does to me. I hope it does to you. Now, how about this guy? This guy's going to subvert, it's going to ruin everything. Unless you think there's shortcuts in life. And there's a lot of people that think there's shortcuts in life. 100 years ago, this was the world's best diet drug. And it works. You know how they always have these internet things about how they tell you, take this pill and burn fat off while you sleep. That's the magic pill, right? The magic pill that lets you burn fat while you sleep, and this one will actually do that. It will burn fat while you sleep. Why? It pokes holes on your mitochondrial membrane. And so let's think what happens to the gradient. Gradient goes down. What happens when the gradient goes down? Electron transport starts running and nothing stops it. It goes gangbusters, running like crazy, because what's the cell trying to do? It's trying to make ATP. But it can't make ATP because the protons are leaking through the hole and not coming through ATP synthase. So what happens to the citric acid cycle? It starts going like crazy. What happens to glycolysis? It starts going like crazy. What happens to your burning of substances? It starts going like crazy. You probably are going to wake up in the middle of the night going, God, it's hot. Because you're going to be running these processes like crazy. That is, if you wake up. About 10% of the people who took this miracle diet drug died. And no, the next question is always, well, what if you just took just a little bit? To which I say, well, what if you just took a little bit of arsenic? Just a little bit. It couldn't be too much. It couldn't hurt. Right? What bullshit. Right? I read in the past month of somebody who recently died futzing around with this stuff. Okay? It's not something to mess with. Things that, and say, well, how long are the holes there? Probably longer than you are. All right? So, beware of miracle diet drugs. Yes? There are research sources of this, and people get those, and then they black market it. Yes? 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 I don't know what else it's called. But anybody who's taking something called a miracle diet drug, I would just say, you know, it's probably not a good idea. Yeah. DNP could be used as a pesticide, yeah. Yeah. The question is, it does only affect you if it's digested, meaning if you take it internally, or if you get it on your skin. You know, I honestly don't know, and I honestly don't want to find out. I get a lot of questions about DNP, and a lot of people are curious about DNP, but it's not something and I just don't know. I just don't know. Yeah. Other questions? Nasty stuff. Okay, but because you now understand something about respiratory control, you can see why this is nasty stuff. You can see why your body heats up. You can see why your body uses oxygen the way it does. And let's put one more wrinkle on this. Okay? I want to talk about electron transport inhibitors. Okay? Electron transport inhibitors. And I thought I had one up here, and I don't see it. Here we go, right here. Okay. All right. There are several things that will block the electron transport system from working. All right? Well, you can imagine if you block the electron transport system, what's going to happen? Probably something pretty drastic, right? Rotenone, up here on the upper right, is a compound that is used to kill insects. And it's pretty effective against insects. Unfortunately, it doesn't have too bad of an effect on us. It's still not something I would take for dinner. What rotenone does is it blocks the movement of electrons through complex 1. It blocks the movement of electrons through complex 1. You can imagine that would have a pretty serious effect because that's where NADH dumps off its electrons. In insects, it's quite deadly. Rotenone is actually a plant substance. It's a natural plant pesticide. And it's very effective on insects. Well, you can imagine if you block complex 1, there are other ways we can get electrons into the electron transport system. Okay? There's another compound called amytal that also blocks the movement of electrons through complex 1. Okay? A nastier compound is actually antimycin A. And antimycin A blocks the movement of electrons through complex 3. You can imagine that antimycin A would be much more poisonous to us than would these two compounds, and it is. There's a little bit of a way to bypass complex 3, but there's not much room to do that. And then you say, well, are there things that block movement of electrons through complex 4? And there are, and you know what they are. One of them is carbon monoxide, something we've already talked about, and I told you how carbon monoxide kills you in 2 ways. One is it binds in place of oxygen in hemoglobin. And second, it blocks the movement of electrons through complex 4. That's deadly. There are other things that block movement of electrons through complex 4. Cyanide blocks movement of electrons through complex 4. There's a compound called azide, A-Z-I-D-E, that blocks movement of electrons through complex 4. These are really, 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 really hard to recover from if you have a chance of recovering. Okay? Let's imagine I took some cyanide. Let's think about what would happen besides the fact you wouldn't have a final exam. Okay? I took some cyanide. What's going to happen? Actually, in our department, we had a graduate student try to commit suicide by taking cyanide. Yes. Okay. Cyanide. Block complex 4. What's the first thing that's going to happen? What's that? Electron transport is going to stop, is the first thing. What's going to happen to oxygen consumption? Think about this. What's going to happen to oxygen consumption? This is one students usually get wrong. Oxygen consumption is going to go down because there's no electrons coming through. No place to put the electrons. There's no electrons coming to put on anything. So oxygen consumption goes down. You slowly stop breathing because you don't need oxygen. What's going to happen to the proton gradient? It's going to fall, right? And nothing's going to build it back up. And then that will ultimately be what kills you, just like suffocation. Let's think about dinitrophenol. You take dinitrophenol, what happens to your oxygen consumption? Through the roof. Through the roof. Because your electron transport system is running like crazy. When you're sleeping your fat off, you're probably going the whole time. Okay? You probably wouldn't sleep through that. But you might not wake up either, so it's a question of which gets you first, right? Okay, yes? Yeah, there are several things that they give to people when they have cyanide overdose, but I'm not going to go into those here. One of the things I'm not supposed to do is talk about medical things, and so I probably shouldn't do that here. Okay. Okay. Well, you guys have been very attentive. I have a video today for you with music. So let me get that turned on. This was a song I wrote several years ago. It's called Around the Nucleus. All right.